Okay, I think we'll start now. So it's a pleasure to introduce Dr. Jiaze Wang. She is a new assistant professor in the School of Earth and Climate Sciences. She got her BS in geology from the Ocean University of China, a master's in marine geology from the Institute of Oceanology, Chinese Academy of Sciences, and her PhD from Louisiana State University. In the letter or the email that she sent requesting information about the Institute and inquiring about affiliation with the Institute, which is the reason that she's providing this presentation, she says the following: as a geological oceanographer, my research focuses on coastal erosion and wetland preservation, coastal ocean biogeochemical modeling, for example, greenhouse gas emissions from deltas and wetlands, and also paleo sea level reconstruction. Specifically, she's interested in studying, one, how coastal hydrodynamics, meaning wave energy, currents, storms, and hurricanes, rework seabed, coastlines, wetlands, and beaches under changing climatic and anthropogenic conditions. Number two, what environmental information has been recorded in sediment deposits, for example, sea level trajectories, ENSO cycles, monsoon intensities. And three, how resilient a coastline ecosystem will be under strong environmental perturbations from both natural and anthropogenic stressors. For example, saltwater intrusion, eutrophication, oil spills, acidification, and global warming. So Dr. Wang is interested in a lot of things, many of which overlap what we do, and we look forward to hearing from you.
Thanks for the introduction. I'm going to share my screen. Can you see my screen? It's empty now.
I can see it.
Okay, how about now?
Yes.
Okay. Yeah, so I have like a very diverse background in research, different research studies, but today I'll just focus on the most recent research work I have been working on. Is about carbon or biogeochemical modeling. So the study area I conducted my modeling studies in Mississippi River Delta, and so it's a very vulnerable coastal system, and there are tons of anthropogenic activities going on there, and is the relative sea level rising rate is super high, and so people are trying to combating with the changing climate. So the carbon cycles, these two most recent studies about carbon cycle in the global scale. The figure on the left shows like the from the land to ocean aquatic continuum, how each system contribute to the carbon cycle. And you can see the estuary and tidal wetlands here. It actually, the right number means the anthropogenic impacts on the carbon cycle. So it actually changing after like present day. This is compared with pre-industry carbon budget. And the figure on the right shows how the global salt marsh changed and in the last 2 decades. And I think Maine is here based on the very coarse color. We actually, our salt marsh doesn't now like change that much. But this is just a salt marsh change. Either it's gain or loss, but for specifically the gain and loss is like an ecosystem shift. It can shift to from a fresh water to a salt marsh. It also can disappear like a mud flat, and it's also when the shifting, if the marsh is gaining, and the upland forest may convert to a salt marsh. So those shift of ecosystem all brings lots of uncertainties to the carbon cycle. In Louisiana is like tons of uncertainties because it's a very heterogeneous system. The soil, we can see the soil types are very different. There's sandy sediment, clay sediment, and also the hydrology, because there's a river and lots of river bifurcations and divergence, and so it's different hydrological basin. And also, like I said, the people are trying to save the land and build lots of freshwater divergence is actually regulate the natural hydrologic zones very fast. So all those can shift can change our understanding, the current understanding of the carbon cycle. So what I'm working on is trying to use numerical models to assess those certain uncertainties. So numerical model. So I use a modified model called P-Flowtran. The model is developed by DOE, a group of people in DOE, but it's a very nice model because it has all different redox network reactions. And in coastal area, it's not like river or ocean. But it is a very complex redox environment. It has nitrogen, has sulfur, has iron, and also has carbon related reactions. So when talking about the carbon cycle or carbon budget in coastal environment, you have to consider all of those redox networks. That is why I build a very complex reactions in the model model framework. And I also want to say this: this model framework is designed for a subsurface environment. It's not for the water. So it includes like methane production and methane consumption, and also sulfate reduction and denitrification. And it also because I'm thinking to look at the methane flux. So I include different types of gas species like nitrous oxide, nitrogen gas, and also hydrogen sulfide and the CO2. I didn't list in this figure, but it's in the model. And the physical, that's the biogeochemical component, but the physical process in the model is it includes advection, diffusion, and bioturbation. We know that coastal or tidal flood is very active environment, and all the worms, the clams, and the bifurcation is very strong, and also gas bubbling. I think Joe I saw Joe here. Joe sent me a video about the gas bubbling or gas escape in Cook Bay, which is very fantastic phenomenon. So yeah, so the model include all those transport process from the subsurface to the surface. And because another feature of coastal environment is the wet dry condition. It can be inundated for a certain time, and it can be explode for another certain time. So in this model, I use a site. It's like the site-specific hydrological data, like water level relative to the marsh surface, which means if the water level is above zero, that means like inundation. If the water level is below zero, that means explorer. Well, because for like a mesotidal environment, it's easy to have like a tidal oscillation function to a model. But for Mississippi, it's different because the water level change is combined with river discharge, wave activities, and also tidal. But the tidal is very small. It has a very small range, like 45 centimeter. So that's why it's better to use a observed set specific water level to force the environment to force the model. And another force in the model is the salinity. Again, it's similar to the water level because river discharge is fresh water, and the waves and tides, they bring ocean or coastal water to the marsh. So it's not like as simple as a tidal function. It can oscillate very differently than a tidal cycle salinity pattern. So I use all the observed data to force the surface hydrology in the model framework. And yeah, so another thing I want to bring out is usually when talk about carbon greenhouse gas in coastal marsh or in other wetlands, the salinity function is very salinity function or salinity relationship with methane flux is very popular, but most of the time the salinity is measured in the water. You can see based on the observation data, the high salinity does not related to inundation, which means if we use a salinity function in a machine learning technique to predict a methane emission, there's a high chance will fail because high salinity those high salinity water is not flooded to the marsh site, which means the greenhouse gas or the related redox reactions is now impacted by the salinity in the water. And so after I build the whole model framework, and I try to sample like as a field biogeochemist, like samples different cores along a salinity gradient. Like this is open water, coastal water. This is a landward. So I picked a few sites and along the salinity gradient and want to reproduce all the relationship we get from the similar to the field data and see whether we can reproduce that relationship from a model perspective. And in this study area, a nice thing is that there are lots of historical methane measurements and also 2 eddy coherence towers, which can be used to validate the model. And you can also see the 5 site I picked is on the right hand side, and they have different bulk density and porosity and also pore water salinity feature. Is where yeah, so here is a few snapshots for the different cores I selected from along the salinity gradient from fresh water to salt water. And this one is like a fresh water, but it has episodic salinity impacts. And from the top to the bottom is pore water oxygen, pore water sulfate, pore water methane. You can see like most of the numerical models, the power it is a powerful tool because it can reproduce the like the high temporal data set, and we can actually see there are some hot moments and cool moments in the high frequency time series data. And you can also see the heterogeneous like in vertical depths and also along the lateral spatial. And for the surface flux produced by the model, I compared with the eddy coherence tower observation data, and the top panel is a ratio between model simulation and the eddy coherence tower. The higher the ratio, the better the model simulation. Well, I have to say that it looks like the model it does not work that well because most of the time it just below 0.2. But again, because it's an eddy coherence tower and the model is configured to simulate the subsurface environment or subsurface methane emission, for if you're familiar with eddy covariance tower, is built like this. It observed the whole ecosystem methane flux, which means that flux is a suite of subsurface methane emission, also the flooding water. We know water also can produce methane gas, right? So it is actually within my expectation, and it is supposed to underestimate the surface flux if I compared with covariance tower data, and also compare, like, see if whether the model capture the seasonal pattern of the methane emission, because we know no matter whether the methane is from the water, coastal water, or from the subsurface, and they all show seasonal pattern, like a high summer season is the peak and the winter season is a trough. So you can see the orange line is the model simulation. If I normalize all the data, it's actually capture the seasonal pattern very well. And this gave me some confidence. And I further compared, because it's a subsurface environment, I further compared the model output with some field measurements which is adjacent to one of the study sites. And one field measurement is the pore water salinity. The triangles are model simulations. I ran the simulation for about a decade, and the field observation was collected like 2006, which is not exactly the time I conduct the model the simulation, but you can see it actually it captured the pattern, the vertical pattern, the salinity gradient. But for the pore water CO2 and the pore water methane measurement, which is very which is I average the model simulation to match with the time of the field data. You can see it actually captured the vertical gradient very well for both CO2 and the methane concentration, which means the model actually can reproduce the subsurface biogeochemical reaction and I mean very well. So after I have gained some confidence about the model framework, I trying to use a data, treat the data like a field biogeochemist, like plot all the data I have, because we know in the model we know the physical drivers, we know the biogeochemical drivers. When we have all the high frequency data, like whether the relationship, whether the model can reproduce or validate some of the field hypothesis, or the relationship between methane and the salinity, or methane and the sulfate. So I plot the surface methane flux against the water level for all the sites I picked, and also plot the surface methane flux against the temperature, and the same. I did the same thing for the subsurface pore water methane concentration with those two drivers, physical drivers, and you can see actually they share. So both the surface flux and the subsurface concentration, they share similar feature, which they sort of increase with inundation and also increase with temperature. And also so for different the relationship between the surface flux and the physical driver are slightly different than the subsurface. One thing here is you see the total, the methane flux actually is centered around the zero water level, which is probably related to the transport processes, like because the model also include the abolition and also diffusion, right? So when the water level oscillates, there's a pressure change. It can pump out all the methane stored in subsurface. And another feature here is that the color bar represent the pore water methane concentration. And if you see the warmer color means high pore water methane concentration. So they the surface, there's like a positive relation between surface flux and the pore water concentration, which makes sense because all the surface flux are from subsurface. And another thing feature is the temperature. Although in the model, the temperature impacts on the biogeochemical reactions has for all sides, they have the same temperature function. But you can see when we plot the methane against the temperature for different sites, they actually show different temperature relation. And that means there's some other things like regulate those relations between the methane flux and temperature, and also the pore water methane and the temperature in the subsurface. And then so this is just a physical driver relationship. If I plot the methane flux and then pore water methane with the pore water chemistry and like other geochemical drivers, because it is so for methane for the methane production or consumption, it is thought that pore water oxygen is a very critical inhibition factor in freshwater environment, and the pore water sulfate is another important inhibit factor in like salty or brackish environment. And this figure is some plot generated from the field data, and the x-axis is salinity. But like I said, the salinity used here is water salinity. It's not it can capture the space like a roughly spatial gradient change, but it cannot be used for a temporal pattern prediction. So I plot all the model simulations and for all the sites. You can see that again it's similar to the last relationship. The pore water sulfate and the surface flux, they decrease along the gradient, like the sulfate gradient, or you can see the salinity gradient. But the oxygen is not, I mean, high oxygen does not quite mean high like low flux. You can see that even when the oxygen is high, and there's still a bunch of methane get out of the subsurface environment. So this figure is trying to say that like in a large spatial scale, there is a gradient, and like from the fresh water and to brackish water and to salt water, and the fresh water can release a much higher methane flux. And if in a sea level rising scenario, and we expect to see the methane emission is reduced if it's converted to salt marsh. But in a like a river diversion scenario, like Mississippi Delta, if you open to a river diversion, and then lots of the brackish marsh or salt marsh can convert to fresh marsh, and then you probably will expect a higher methane emission from that new created freshwater land. Well, this means like if we preserve land, and we probably also get some high methane emission from the new land. And another, well, this figure is another busy figure, because like I said, the model include gas transport from subsurface to surface, and the gas transport, they have diffusion and ablation, and I was like, oh, can I distinguish those two fractions, like how much flux is contributed by diffusion, because we know diffusion is gradient driven. If you have a high methane concentration in subsurface and you are expecting high diffusion flux, but ablation is another, it's like a chaos process, and high methane concentration does not quite mean high ablation flux, and also ablation can happen like very episodically, and is just very difficult to capture. So because it's because the model can include that, so I can actually separate those two components. And this figure is trying to show you that the ablation flux actually contribute. Well, it is episodic, like the small figure here, but it can contribute to very large fluxes to the total surface flux. And diffusion is just here. It is constant contributing to this total flux, but it's very small number. The cumulative, but because the ablation is very episodic, although it can contribute like a large flux to the total gas flux, it actually only contributes to 20% of the surface flux in the model. And also you can see for different color means different marsh sites. Like for brackish marsh, they actually can produce similar ablation methane flux with a freshwater marsh. And then, like a detailed look about the ablation process is, and so the frequency, because it's episodic, and the model actually can take a look about how frequent ablation can happen for different sites. Well, this figure just show you the pore water ablation frequency does not quite related to pore water concentration, because in the model I also include different gas species like CO2, nitrogen oxide, and nitrogen gas. All those gas contribute to ablation gas can include all those gas species. So that's not like the frequency, like a high methane concentration does not mean it will contribute to the ablation like a frequency. But when the warm color indicate high pore water methane concentration in the subsurface. When the pore water methane concentration reach to a level about like in the model 175 micromole per liter, it actually can like increase the ablation frequency a lot. But below this threshold is not highly related to the frequency. But what because the model has all other gases, and the methane ratio in the bubble gas is actually related to the subsurface methane production. You can see it actually increased up to like a less than 20% in composition in the gas bubble. Well, that 20% means that other is like it's saturation, gas saturation threshold, because there are other gas also produced in subsurface environment. Yeah, so this is just some detailed information in the model, which I personally want to share here. And so so far the model, some capability of the model is like it has physical driver, can we can observe the physical driver for the methane reaction. We also can distinguish the geochemical driver like salinity or eutrophication, which I didn't show in this slide because the model actually have include the nitrous oxide, which I do have the data. I just didn't plot here because it's too much. And also the model also include a wind effect, which I also is there and is included, but I didn't show in this presentation. So this is a bunch of gases like greenhouse gas produced in the model, and also other gas, and also benzene exchange, like how the DOM exchange with overlaying water. It's all the information can be produced by the model. But again, it's a biogeochemical model, and it like all other biogeochemical model, it has its limitation. So it represents all the biogeochemical reactions with a fixed rate constant, which is far away from the reality. And so in reality, we have all different microbes, and they have different reaction rates, they have different functions, they adapt to the changing environment, like changing temperature or changing salinity very fast. And they just well, from a community scale, they probably you probably can see the same thing, like the methane emission is still the same, but from a biological scale or the macro species scale, they probably, the species probably change, like have a community dynamic structure, changing environment. So that means the model actually means a diverse microbial community. And that's lead to my next topic, is move forward to a more metabolic complexity model. So the goal of the following work is trying to represent microbial species and their gene functions and also community structure explicitly, because actually, so most of biogeochemical models, if they roughly do a good job on reproducing the large spatial pattern of like nitrogen, oxygen, all those things. But the biology, the dynamic of the biology is not well presented or represented. So, well, it's okay if the environment is a stable environment. We can there's no so we can still use that type of model, but in a changing environment, so if we are like a increasing temperature, or the diverse microbial community may may shift. So like some dormancy functions or some dormancy microbes, they may wake up and trying to regulate the biogeochemical environment. But right now our models cannot do that. So that is the motivation of this type of work. How I did it. So it's a very complicated process. I try to make it as simple as possible. And it's again, it's like I'm trying to build a artificial microbial community, which like each metabolism represented by each functional genes. And we have we can build a very complicated redox network, but we cannot build like a diverse microbial community. So what I did is trying to nest those complicated redox network into an artificial microbial species, like make it as diverse as possible. And which means you can think about like each microbes in this community, they actually function like they can function like a complex networks. And well, the test here is just trying to say the capability of this framework. Yeah, so this is a model case study with that type of model. I applied that model to Gulf of Mexico. I introduced that model to a physical ocean model environment, and this is Louisiana coast, and trying to invest how those microbial species responds to a deep water horizon oil spill. So in the model, I put about 58 microbial species. Each species, they have several genes. Some of them have 11 genes, some of them have 7 genes. And so each genes represent one type of metabolism. Some of them some genes is related to light inhibition, and some of them related to carbon degradation, and or some of them are related to nitrogen fixation. But because like all other ocean model, it use nitrogen as a currency for all the biogeochemical reactions, and the carbon is actually represented by a Redfield ratio. And the plot here is the day after the oil spill, and the color here means the gene copies in the model. Like you can see this is a representative gene about ethane degradation. You can see the growing of the gene copies right after the oil spill. And so again, I did the same thing. Like if I'm a field microbiologist, I probably want to sample the microbes and see how whether the artificial, like how the artificial microbes respond to the change of the environment. So I sampled one species, microbial species, which bear a gene with ethane degradation, and I plot the simulated transcription for different genes in that microbes. You can see that like the different, like the interesting is like different functions actually respond or shift during the oil spill, which is cool. Like I think that's like in the lab, and our genome people probably see the similar things. And so this is under SIP condition, which means that before the oil spill, there's natural SIPs flux into the system, and you can see the microbes. This microbe respond to the oil spill. I also did the experiment is like, if there's no SIP condition, and whether this microbe respond to the oil spill the same way it does like with a SIP condition. So, okay, same bug, but different, same microbe, but different metabolism respond, which is like a simulated gene scale response. And then I was like, okay, can I compare those, compare the transcript level of the same microbe in different conditions. You can see the blue color is a no SIP condition, and the orange color is a SIP condition. I mean, they actually have different pattern, and the transcript or the metabolism is respond differently, even if it is the same microbe. And then I look at the data with, like in a mall community response, and for community response, you would want to check the meta transcript, like the whole community, pull out all the ethane degrading gene together and from different species, and see how it regulates the spilled oil or the changing hydrocarbon concentration. And this plot is just showed for different environments. They have different meta transcript pattern, and they regulate. You can see it, about geochemical will deregulate the concentrations of the hydrocarbon differently. And then I check the geochemical result, because in the ocean we care about nitrate, we care about oxygen concentration. With a diverse microbial community, whether those communities should change the geochemical, the system level geochemical pattern different or not. So this is the reason why I plot those two figures, and the left figure is about nitrate concentration for different condition, and the right figure is about the oxygen concentration. Again, they have different pattern than the micro. So same microbes respond to the same oil spill or same pulse disturbance differently, and because the precondition is different, and they have the community. So microbial community cultured in different conditions work on the pulse disturbance by Deepwater Horizon also differently. This is trying to say the response from gene scale to community scale to system scale actually is very dynamic. They are very different. Even you have the similar metabolism but different preconditions. Yeah, and then that's ocean system. Then was in the subsurface, like a soil or sediment environment, the microbes are I don't know whether it's much more diverse or not, but it's very diverse. So I was thinking, can I do the similar thing and make it more suitable for a subsurface environment? And then I create a similar framework and perturb the system using a flooding year and the saltwater intrusion year. Which flooding year means like the salinity does not change, but only the water level or the inundation depth is increased. Saltwater intrusion year means like I increase the salinity, it's like a sea level rise, and see how those simulated microbes respond to those disturbances. And in this framework, because it's a test, it's just for fun. So I put 15 microbes in the model, and here are some examples of the microbes and the function, the metabolism or functional genes included in the model. So each microbe, they bear 2 genes, and some gene, one gene is about sulfate reduction, or they can represent other like acetate oxidation, and the other gene is about nitrification or soil organic matter decomposition. So I release those microbes in the model and see how it responds to the environment. And the top panel is soil, is the top soil, and then all the way down to the bottom sediment or soil. You can see different color means different microbes. Even they have so every so far every microbes, everything is everywhere. So in the first layers, there are total of 15 microbes. In the bottom layers, also 15 microbes. Well, you can see that even the same microbes, they actually respond. Like here, the microbe 11, it responds to the environment differently. Like in the top layer, it grows increase very fast, and in the bottom, it's just reached to its limit very very early stage. And then I checked their growth rate based on the model simulation. Yeah, well, it just matched with the growth, like the biomass growth, and they, even they are the same microbes. They respond to the they have different response in different depths. Well, it makes sense because the different depths have different biogeochemical environment. Like the sulfate is different, the soil organic matter is different, and all like oxygen is different. So, and then I checked the gene transcription in the produced by the model. Like one transcription one perturbation is the is fresh no, it's flooding, and the other perturbation is saltwater intrusion. And you can see the here, I mean, like the shift, like for microbe 1, when there is a flooding, and it actually shifted to a sulfate reduction, because the flooding in the model I configure is not freshwater flooding. It's the same salinity but increase the water level. So it shifted from a SOM soil organic matter decomposition pathway to a sulfate reduction pathway, because it's more the perturbation, the disturbance is more favorable for that metabolism, and it can grow faster or can adapt to the changing condition. So yeah, so it adapts to the changing salinity or changing water level. And same here for microbe 14. Although because for these two for microbe 14, the two pathways, one is about sulfate reduction, the other is about methanotrophy. So it shows that the sulfate reduction is actually a very the growth of this microbe benefit from the sulfate reduction from the beginning. And when there's a perturbation, it create a more provide a more juicy food for that pathway. So it just respond to that very like higher degree than before. Yeah, so I mean the subsurface simulation is just a like a test. It's not based on any lab data, or it's not compared to, or with any field observation. So there are lots of room for that work to be improved. And here are some thoughts of my next step about that model, and then that's conclude my my talk. And also I want to thank all my collaborators. And at the very end, I want to share some photos I took in the field, which the sea level rise here, the marsh, the change of the marsh area or salt marsh can happen very differently. Like here is like tidal channel expansion, but in this figure, it's like the interior degradation can happen. So all those processes are very different, and they have different biogeochemical consequences. And this figure is about a human activity. On the left side is a restored, I think it's a Dutch Unlimited. It's like a freshwater restoration. But on the right is the regional salt marsh area. And so you actually can expect the restored freshwater wetland can probably becomes like a methane hotspot in the future if we continue doing that. And here is a figure while we're hiking in Tennessee, I found like a very cool mining, like a carbon storage in the mountain. And I just thought it's very cool to see that in they expose the carbon storage in the field, and it just like the green trees may become those carbon in the future. Anyway, thanks.
Thank you, Jiaze. Questions from the audience?
Yeah, I'll ask a question, Jiaze. When you were talking about methane fluxes, and I'm assuming these are under ordinary, regular conditions. What about in a large hurricane, a big inundation event with salt water probably coming in, but maybe even a great deal of fresh water? Would that be a major methane release time?
I had a simulation about that, because I'm also interested in see how the methane changed after a big hurricane, like a long-term inundation. It actually so I didn't show it here. It actually it's a combination about inundation and the pressure change. So during a hurricane, there is salt water is the magnitude actually is lower if it's a fresh marsh, but if you compare with a salt marsh, it's not that low, because there's a window. It kind of the high water store like favored some of the methane production and the store on the subsurface. When the water retreat, it just like the pressure change, it just come out very fast. It's like create a large scale of like pulse flux, but it doesn't last longer. Yeah.
Thank you.
Any other questions? I have a quick question. If you could go to the last data slide. It's a very minor question. It would be number 23.
23.
So if you look at the soil depth 1 centimeter, the second one from the right and the third one from the right have a big drop around 8 years. It seems very anomalous. What do you think's going on? Maybe you explained that and I missed it. What do you think is going on?
So I did, because the model, because I thought the model is configured with all the micro biomass. There is a community pressure. They cannot grow as much as they want. And there's, if the biomass increase a lot for that certain species, I have to kill them. Like the model actually is more like a pressure. It just died, or you can, well, I think I also add the virus killing them. So it's just a job based on the like other species, they probably become dynamic and then just kill them.
And the other question, I assume that the variability throughout most of them is some sort of seasonality. Is that correct?
Yeah.
It seems as if it dies off super fast and comes back on super fast. Is that the way you would expect that to work? Such a sharp drop and then a sharp rise?
Say that again? What?
Well, looking at, for example, at soil depth 6 centimeters, the first one from the second one from the left, or any of those, sorry, the other way. Right, sorry, yeah. I assume that up and down, the 6 centimeter. I assume that the up and down is some sort of seasonality, right?
Yeah, that's yeah.
And why does it go up so sharply? And it's as if everything forms and then literally dies very fast and then comes back very, very fast. Is that realistic, do you think?
So I don't know it's realistic or not, but I know why it is in the model.
Mhm.
Because this is based on this is a biomass change. No, not biomass change. Is a transcription. Is the
Mhm.
pathway, how the pathway respond to the change of the like, so I think this one is nitrification.
Mhm.
So it just because the way the model works, because it has to function to maintain its life for that microbe. So it just keep going on, but also when it keep the metabolic function is expressing itself is based on whether the nitrification or the nitrate concentration is high or low, and whether there's competitors. So that's another reason it expressed higher or lower, and also based on its total biomass.
Okay, wonderful. Thank you.
Yeah.
Are there any other questions out there? I don't think I see any hands up. Well, thank you so much, Jiaze. That was a wonderful talk. You gave us an awful lot of information, and we look forward to talking with you more. Thank you so much. Have a good day.
Thank you.